Good morning. So in part 6 of chapter 4, we are going to be getting into the concepts of protein stability and folding. So at the core of this conversation are going to be non-covalent interactions. In particular, we're going to be focusing on interactions like the hydrophobic effect, electrostatic interactions, hydrogen bonding. And we're going to talk about how those non-covalent interactions come together to stabilize the structure of a protein. We're also going to talk about the order of events. So when you have a newly synthesized polypeptide or protein, straight off of the ribosome, straight out of the oven, how does it get from that largely unstructured state to the folded, structured, potentially globular, three-dimensional structure that we associate with the active form of, say, an enzyme? So how do you get from point A to point B? We're going to be talking about how that happens here.
As we move through the major non-covalent interactions that stabilize protein three-dimensional structure, we're to some extent going to do so in order of decreasing priority. So here we're going to start with the hydrophobic effect. The hydrophobic effect is the major determinant of protein structure. When we talk about globular proteins, we talk about them with the assumption that they have an interior, solvent inaccessible, nonpolar core and a hydrophilic exterior. So that is the consequence of the hydrophobic effect. That structure forms as a consequence of the hydrophobic effect. Now, let me give you a brief refresher. What is the hydrophobic effect? We talked about it before, but here we're going to talk about it in a slightly different context. So if you recall, this is a figure that you have seen before, but if you recall, the individual hydration of nonpolar molecules requires that water molecules basically move out of the way to create volume, to create space that can be occupied by each individual nonpolar solute. So this is important. If your nonpolar molecule, and this is true for any solute for that matter, but if your nonpolar molecule is going to be soluble in water, it has to occupy space, volume, within that aqueous environment. So those waters, even though they aren't really interacting so much with your nonpolar molecule, they still have to part. They have to move out of the way to accommodate your nonpolar molecule. Now, if you do this individually for every single nonpolar molecule, you will have a greater surface area than if those nonpolar molecules just sort of cluster together. Remember here, surface area is proportional to the number of water molecules that have to become ordered to basically allow for the nonpolar molecule to exist in that aqueous environment. So the number of water molecules that are required to accommodate the individual hydration of nonpolar molecules is more than in the case where your nonpolar molecules cluster together. Now this is important because the more water molecules that have to become ordered, the greater the entropic cost that is paid by the system. Those water molecules are giving up entropic freedom in order to accommodate the nonpolar molecules that are present in solution. So if we can minimize that entropic cost, we're in a better position. And that is exactly what is happening over here. When our nonpolar molecules cluster together, there's still an entropic cost. That is the entropy goes down for the entire system, but that cost is less over here relative to the case of individual hydration of nonpolar molecules. Okay, so I've given you the refresher as to what the hydrophobic effect is, but why are we talking about it now? Well, that is because the hydrophobic effect is the major determinant of native protein structure. Remember, I just told you that for a globular protein, your interior, your solvent accessible interior for your globular protein structure is going to be nonpolar. So what happens is your nonpolar side chains, think about things like amino acids like alanine, leucine, isoleucine, they are going to aggregate in the interior of your protein structure. And this is the consequence of the hydrophobic effect. We're trying to minimize the required ordering of water molecules. So by making those nonpolar side chains largely solvent inaccessible, we don't have to worry as much about that entropic cost that would be associated with the hydrophobic effect. So here, the aggregation of nonpolar side chains in the interior of a protein is favored due to the increase in the entropy of water molecules that would otherwise have to form ordered cages around your hydrophobic groups. So basically, we are adopting a protein structure that crams all of your nonpolar side chain, all of your nonpolar amino acids into the protein interior, thereby making those functional groups solvent inaccessible. And for the system as a whole, you don't then have the entropic penalty that would be required to form those cages, those hydration shells around your nonpolar functional groups. So the hydrophobic effect is really very, very important. Now, before I move on, there is one thing I want to add here. Protein three-dimensional structure is stabilized by non-covalent interactions, the hydrophobic effect being the most important of the non-covalent interactions. But, on average, the free energy to denature a protein is approximately 0.4 kilojoules per mole per amino acid residue. So as an example, for a 100 amino acid long protein, so if we have 0.4 kilojoules per mole per amino acid, that means it would take about 40 kilojoules per mole to denature a 100 amino acid protein that is structured. Now, 40 kilojoules per mole, that is not incredibly stable. That means we have to put in almost the equivalent of 1 ATP in terms of ATP hydrolysis energy to unfold that protein. So when we talk about proteins being more stable and non-covalent interactions leading to their stabilization, that is something that is highly relative. And what I mean by that is, yes, the folded state, the globular native protein structure is the lowest energy, most stable state, but it's still to some extent a house of cards. You put in a little bit of energy, maybe you raise the temperature a little bit, or you perturb the system just a little bit, and that's all it takes to cause the protein to lose structure and denature. So just remember that when we talk about protein stability, yes, the non-covalent interactions that stabilize that protein structure lead to greater protein stability, but it is still a very tenuous situation in that it would take very little to push that protein into the denatured state. So it's a delicate balance. Okay, so if the hydrophobic effect is so important, let's talk about that a little bit more. When we look at individual amino acids, every amino acid is generally classified as being nonpolar or polar charged, polar uncharged. We've talked about these classifications. And so what I'm about to tell you should not come as a shock. When we write out the primary sequence, the amino acid sequence for a protein end to end, so N-terminal end all the way down to C-terminal end, every amino acid can be quantitatively described as being nonpolar or not, or polar. And the way that we do this is using a parameter called the hydropathy, or hydropathy index, or some iteration of the term hydropathy. So when we look at all 20 amino acids, on the right-hand side here, you'll see a table that contains all of this information. Every amino acid side chain, and here remember we are considering only the side chain because that is what determines whether an amino acid is overall polar or nonpolar. You'll see that every amino acid side chain has a hydropathy index associated with it. Okay, so the hydropathy index, what is it? Well, it is a way to express the combined hydrophobic and hydrophilic tendencies of an individual amino acid. So the more positive that hydropathy index is, the more nonpolar the amino acid side chain is. Okay, so something that is more nonpolar is much more likely to reside on the interior of a protein, a globular protein structure. So you'll see isoleucine has a hydropathy index of 4.5, valine 4.2, all the way down to alanine, which is 1.8. All of these should not come as a surprise to you. Alanine, methionine, cysteine, phenylalanine, leucine, valine, isoleucine. These are all amino acids that we would consider to some extent be nonpolar. Cysteine is kind of the wild card here. It's not terribly polar, but it is classified as a polar amino acid because under some circumstances it can form hydrogen bonds. But overall, based on hydropathy, it's pretty nonpolar. As we keep working our way down the list, you'll see arginine at the other end, minus 4.5. So your negative values are amino acid side chains that would be considered polar and are likely to be solvent accessible. So when we look at the entire protein sequence, we lay out an amino acid sequence for a protein from end to end. What we can do is actually plot the hydropathy index for each amino acid end to end. And in reality, this is not as straightforward as it's being presented. I want you to understand that there is some averaging and algorithms go into this, but in a simplistic perspective, if you plot hydropathy index for each amino acid, what you can do is actually identify regions of your protein that are more likely to be solvent inaccessible in the protein interior or solvent accessible, which would be something that's more likely to be on the surface of the protein. So as we walk around, from end to end here, you'll see, you've got some positive values here, but just a small stretch. But then we sort of work our way down here, and you'll see, there's a whole bunch of stuff right here that have positive hydropathy indices. So this bit is probably buried away on the protein interior, so it's probably solvent inaccessible. And then you'll find a stretch where you've got some negative values. This is probably something that is solvent accessible. It's probably on the surface of the protein. Then you've got another stretch that's has more hydrophobic in nature. And on the end here, there's a bunch of stuff that is more hydrophobic in nature also. So from the hydropathy plot, which is what I've shown you here, you can identify regions of your protein that are more likely to be buried on the protein interior, and conversely, you can identify parts that are more likely solvent accessible on the surface of your protein. The next most important non-covalent interaction to contribute to protein stability are electrostatic interactions. Now, as I'm talking here, I want to make it very clear that in this context, what I'm actually talking about are hydrogen bonds. So in most cases, when we talk about electrostatic interactions in proteins, what you're actually talking about are hydrogen bonds between charged amino acid side chains. So for example, here you'll see the primary amine side chain from lysine interacting with the carboxylate from glutamic acid or glutamate. So here, this is technically a hydrogen bond, but at the end of the day, the hydrogen bond is a dipole-dipole interaction, and that is driven by electrostatics. So we're going to consider this as an electrostatic interaction. Now, hydrogen bonding is incredibly important to the stabilization of protein structure. But, they really only contribute in a minor way to overall protein stability. Now, this is not a statement that is meant to minimize the contribution of hydrogen bonds, but when comparing against the hydrophobic effect, hydrogen bonds don't contribute nearly as much. So that is something that we have to really consider here. Now, the real contribution, not just to the delta G that would be used to describe stability, but the real contribution of the hydrogen bond is to guide the actual shape of your protein. So as you'll see here, the hydrophobic effect stabilizes a small number of conformations. So if you want to sort of consider what we're talking about here, imagine the situation where you have the newly synthesized protein or polypeptide. It's been freshly made, freshly synthesized off of the ribosome. At this point, it's largely unstructured. So to make an analogy here, imagine like a block of clay. Now, the hydrophobic effect is going to lead to the formation of some structure. So your block of clay adopts a small number of shapes. You can sort of make a couple of different shapes out of that block of clay. But then, in the context of the protein, hydrogen bonding starts to occur. And so you start fine-tuning your tertiary structure. Now, you've started, sort of carving away at your block of clay, but it's not quite where you want it to be yet. But it's, the hydrogen bonds sort of stabilize intermediate structures. Not your final native structure, but intermediate structures. Okay, so hydrogen bonding here is important because it's leading you towards that unique lowest energy native structure. Now, these hydrogen bonds that we're talking about, there are specific names that are used to describe them. In particular, something like this, a lysine with a glutamate or an arginine with an aspartate, something like that. Two ionic groups of opposite charge interacting. We commonly refer to these as ion pairs or salt bridges. You'll hear me use the term salt bridge a lot. These are just another example of hydrogen bonding interactions that, again, fall under the electrostatic umbrella. Now, I told you a minute ago that these interactions contribute very little to the overall protein stability. That is, when you calculate the delta G that would describe protein folding, the difference between the unfolded and folded state here, numerically, these contribute very little. I just told you they help to fine-tune the tertiary structure, so they are important, but numerically, they don't contribute a whole lot. The reason for that is really something that we have to consider, we have to consider the case of the unfolded versus folded state. So in the unfolded state, all of your side chains have entropic freedom. They're moving around. They're hydrated by water. But then when they form the salt bridge or the ion pair, what happens is those side chains have to undergo desolvation. They have to shed those water molecules to facilitate interaction with something else. They also give up that entropic freedom. So there is actually an entropic, let's just say more broadly, a thermodynamic cost to forming the salt bridge. So there is a loss of entropic freedom. There's a loss of solvation energy that comes with forming the salt bridge. But they still contribute overall to the stability of the protein, which is why they happen. So they're important to overall stability, in terms of fine-tuning tertiary structure, basically leading you to the lowest energy state. But there's a compromise in doing that. The next interaction that we will talk about is, in fact, not a non-covalent interaction, but is instead a covalent interaction. These are disulfide bonds. So we've talked about the disulfide bond before. This is when the side chain of cysteine forms a covalent bond. It undergoes oxidation with another cysteine residue that is located nearby to give you the disulfide bond or disulfide bridge, as you'll hear me refer to it. This was particularly relevant when we talked about the structure of alpha keratin. You'll remember cysteine became cysteine, individual cysteines underwent oxidation to form disulfide bonds. So disulfide bonds are very important, but there are some caveats we have to talk about here. So having two cysteines adjacent to one another does not guarantee that you're going to form a disulfide bond. For intracellular proteins, disulfide bonds are somewhat rare because the cytoplasmic environment is highly reducing. So in that cytoplasmic environment, if you have two cysteines adjacent, you have a very reducing environment which favors this state, the individual cysteine side chains that are not in a disulfide bond. Okay, so that is not to say that disulfides can't form in the intracellular environment. Because of the non-polar nature, the hydrophobic character of the disulfide bond, you will sometimes see disulfide bonds occur in the intracellular environment. And when that happens, very commonly, you'll find the disulfide bond to be in the protein interior. So in that non-polar protein interior, you'll often find disulfide bonds. Now, more commonly, you're going to find disulfide bonds in extracellular proteins. Most disulfide bonds occur in proteins that are secreted from the cell into the extracellular environment, which is in fact a more oxidative environment. The protein in the extracellular environment, it needs to find a way to stabilize against the many hostile stressors that will present. So think about, in an extracellular environment, you have uncontrolled temperature and pH, you have chemical stressors, you have all kinds of things. So many extracellular proteins do, and rather are found to have more disulfide bonds than intracellular proteins. Now this is interesting because when we talk about the order of events here, the hydrophobic effect is going to sort of guide your initial structure. The next step is going to be the fine-tuning of that tertiary structure via hydrogen bonding and electrostatic interactions. And then, as a terminal step, disulfide bonds start to form, and this is important to more or less lock in a particular conformation. So disulfide bonds, when we talk about the order of events, disulfide bonds are often going to be one of the last interactions that form. And this is, again, just basically a way to lock in a particular tertiary structure or conformation. An additional scenario that we need to talk about is that of metal ion stabilization of protein structure. So it is not uncommon to find that metal ions will stabilize protein structure as well. There are, in fact, certain motifs or structural arrangements that position amino acid side chains, particularly cysteines and histidines, to be arranged in such a way that you can coordinate a metal. So the example I'm going to give you here is the zinc finger. Zinc finger domains are very common in nucleic acid binding proteins. They're really very common and support the structure that is needed to bind DNAs and RNAs. So in those structures, you'll find something that looks like this. In a lot of cases, you have an alpha helix that feeds into some beta content. So here you have a beta strand and another beta strand on the backside. But what happens here is you have on this alpha helix two histine residues. These could be cysteines in some cases, but two residues that are on the alpha helix positioned to be on adjacent turns. On the beta strands in the loopy structure here, you'll see two cysteines. Cysteines are represented here in yellow, and you'll see the blue represents the nitrogen of the imidazole side chain of histine. And ultimately, they're going to be positioned in such a way to coordinate a zinc ion. So zinc, it is a metal ion that prefers to be in a square planar arrangement. So that means you have four different things around it in a square planar sort of geometry. Now here, the cysteines and the histines form that square planar geometry. This is interesting because what the zinc actually does is basically pulls this alpha helix and keeps it bridged alongside the beta content here. So if this zinc were in here, this alpha helix, because there's a flexible connector, would sort of be flopping about over here. But the zinc kind of pulls it in and functions to cross-link or bridge the alpha helix with the beta strands over here. So the zinc helps to sort of keep everyone together. Now this is not specific to zinc. You'll find other metals do the same thing, but zinc is a very common example of a metal that stabilizes protein structure. Before we move on, I want to show you some real-life actual examples. What are some examples where these non-covalent interactions and covalent interactions in the case of disulfide bonds, where might you see them? So here, I'm going to throw up a figure for you. So here, I have 4 different examples highlighted. In each case, I have the protein structure shown in cartoon view or ribbon view alongside the protein topology. So we haven't talked a lot about protein topologies, but this is just a way to represent the secondary structural arrangement for a protein. So in this case, defensin. For those that are interested, this is a antimicrobial protein that is secreted from grapevines to prevent from bacterial infections. So this is a pretty cool example, but it's a relatively simple protein. You have, if you look at the structure here, a small beta sheet, 3 individual beta strands that are oriented anti-parallel to one another, and then a single alpha helix. If you look at the topology, you'll see here you have your anti-parallel beta sheet, 3 strands, anti-parallel, and you have your alpha helix. Okay, so here you don't really know what is keeping this together. I mean, the beta sheet, you probably have hydrogen bonding between the strands like we talked about, but that doesn't tell you how the alpha helix interacts with the beta sheet. Why is this alpha helix held in the position that it is? Okay, so the same is true for any other protein that you look at. If you look at this zinc binding domain, it's a little bit more clear. You have a zinc ion that holds everybody together. In this case, for the zinc binding domain from E. coli clip X, there's a zinc that is being coordinated by 4 different cysteines that are shown in yellow here. Your alpha helix is held in place by this zinc interaction relative to your beta strands. In the case of the human BAZ1A bromo domain shown here in part C, you've got a bunch of alpha helical strands that are held together also. You know, based on the backbone hydrogen bonding, how the individual helix forms, but you don't know much more. And there's lastly a more complicated case here. But okay, so I'm not really going to spend a whole lot of time on part D here, but what I want you to understand is that each one of these cases that I'm showing you is stabilized by a different kind of interaction. So that last example of the part D here, the AAA binding domain, it's a more complicated structure, but if you look at it, you'll see that you have 3 layers. There's a layer of alpha helical structure, a layer of beta structure, so there's a beta sheet here, and then another layer of alpha helical structure. If you look at the interface between the layers, you'll see an enrichment of nonpolar stuff. So in particular, in this case, you have, like here, there's a phenylalanine, and here's a tryptophan. So those are relatively nonpolar, and you probably have pi-pi stacking here. In this case, there is an arginine, which is polar, but that arginine is interacting with the pi electrons of a tyrosine. So you have a pi cation interaction. On the backside, you'll see some nonpolar stuff. So your layers are formed as a consequence of the hydrophobic effect. So that globular structure, and yes, this is roughly spherical in structure, that globular structure in this case is supported by the hydrophobic effect. Now, in that case of the bromo domain that I just showed you, the alpha helical segments are held in place due to hydrogen bonding interactions between adjacent alpha helices. So we started off with an example of the hydrophobic effect. Now we have a case of hydrogen bonding interactions holding everyone together, sort of fine-tuning tertiary structure. In the case of defensin, those secondary structural features are largely held in place by disulfide bonds, disulfide bridges. This is an extracellular protein, so you do see more disulfide bridges in defensin proteins relative to intracellular proteins. So here you'll see three separate disulfide bonds, one of which holds the alpha helix in place relative to this beta strand. The last case is that example of the ClipX zinc binding domain, and you have a zinc ion that holds your alpha helix in place relative to everybody else. So what I'm trying to really show you here is that your hydrophobic effect, hydrophobic interactions, help to form that initial structure, that initial tertiary structure. But that state without other interactions does not look a whole lot like your final native state. It is through these hydrogen bonding interactions and disulfide bridges, metals, these really lead to the fine-tuning of your terminal protein structure, your native protein structure. Now that we've talked about the non-covalent and covalent interactions that stabilize protein structure, let's talk about what actually happens in the cell. How do we go from unstructured polypeptide to folded functional native protein? Okay, so let's start here. We're going to start at the stage of the ribosome. So your ribosome, it's bound to some mRNA. It's chugging along, synthesizing a new or nascent polypeptide or protein. And when it's done, that polypeptide is going to leave the ribosome. So at this stage, it's largely unstructured. The polypeptide or protein, it's, in my mind, basically a floppy noodle that has no real structure. So how do we go from there to our functional protein? So what's going to start happening? First, due to the hydrophobic effect, you're going to see the formation of a crude tertiary structure. Then you're going to start getting, adopting different conformations that lead to different hydrogen bonding and arrangements. So these are going to lead to folding intermediates. These folding intermediates, they contain structure. They're structure. They have a lot of your secondary structural elements, but your final tertiary structure is not quite there yet. So in some cases, you can go from folding intermediate to native protein without any assistance. For small proteins, this is generally the case that they can fold into their native structure independently. They don't need help to do so. But larger proteins may have trouble doing that, and so may need some help. So there are these enzymes that are called chaperones that come along and help the folding intermediate adopt the native protein structure. So chaperones are really important for ensuring that proteins find that native conformation that leads to the functionally relevant state. Now, one thing to take note of here, the interactions that largely lead to your native protein structure, with the exception of disulfide bonds, are non-covalent interactions. So non-covalent, what this implies is reversibility. You have interactions that can form, but they can also unform, and this is a reversible process. So to go from the folding intermediate to the native state, that in most cases is a reversible process that, if stressed in some way or another, then the native protein can readily reverse back, can unfold back to that intermediate state. So ideally, what would happen is your nascent polypeptide would start adopting folding intermediates, would start sampling different conformations, where hopefully one of those conformations is going to lead to the lowest energy native state. Okay, so protein activity, enzyme activity. I view this a lot like a light switch. When you need a particular metabolic function or enzymatic activity, you flip the switch and turn on a particular gene. So gene expression goes up. You start making more mRNAs, which leads to higher levels translationally of protein. You start making more protein once you flip that switch on. But at some point, you're probably going to exit the room. And you're probably, I hope, going to turn the light off when you leave the room. This is a lesson that my 4-year-old has not quite learned yet, but hopefully one day he will. When you leave the room, you turn the light off. To make that relevant here, you have turned the gene on. You turn it on because you need the function of the enzyme that is being produced, but you may not always need that. So once you turn that gene back off, you stop expressing that protein. Really, that protein at that point has served its useful purpose. It has lived its life and it's done its thing, but you don't really need it anymore. So once you turn that gene off, stop making protein, you need a way to regulate the enzyme that has already been made. So what you're going to do is take that, and in most cases, you're going to degrade it. So that native protein is degraded into peptide fragments, which through the action of other exopeptidases, those peptides can be broken down into individual amino acids and recycled back around to make more protein. So this is, the life cycle of the amino acid. Okay, so what I just described to you is the situation where everything goes perfectly. That is not always the case, though. If it worked perfectly, we wouldn't have disease. So, okay, what happens when things go wrong? Folding intermediates. By definition, these are intermediates. Remember, in the final state, the final folded state, the native state, you're going to have a nonpolar interior and a polar exterior. Folding intermediates, on the other hand, because they are not quite to that native state, may have nonpolar residues that are, in fact, solvent accessible. So, we're not perfectly structured yet, so we still have exposed nonpolar surfaces in our folding intermediates. Occasionally, what can happen is a folding intermediate bumps up against a neighboring folding intermediate where they both have exposed nonpolar surfaces. Because of the hydrophobic effect, those nonpolar surfaces will sort of cluster together and potentially lead to the formation of these aggregate structures. Aggregates can be amorphous, so they lack general structure. They can form oligomers that are ordered, whether it be like this, or in the case of amyloid fibrils, be ordered in a different way. In either case, all of these aggregates can be very toxic to the cell. So when we think about protein aggregation disease, things like Alzheimer's, Parkinson's disease, Huntington's disease, these are all protein folding diseases that form due to unwanted aggregate formation that is toxic to the cell. So wherever possible, what we're going to do is employ a specialized class of chaperones that facilitate disaggregation. These are often referred to as disaggregases. So they come along and they recognize these aggregates, and imagine this amorphous aggregate as sort of a ball of yarn. These disaggregases bind to these amorphous aggregates and start sort of tugging on that ball of yarn. Apply some sort of pull force to the ball of yarn. And eventually what's going to happen is your ball of yarn is just going to sort of fall apart. When that happens, hopefully what we're going to see is a transition from aggregate state back to folding intermediate state. And at that stage, other chaperones can bind to the folding intermediate and try to promote the native structure once again. Okay. In some cases, folding intermediates do not aggregate, but can instead, remember, the folding intermediate is sampling different conformations that differ in overall free energy, hopefully towards the goal of moving down in free energy towards the lowest energy state associated with your native protein. In some cases, you can sort of get stuck in these locally stable conformations that are considered to be misfolded. Misfolded proteins can lead, they can aggregate as well. They are the subject of a lot of study, these misfolded proteins are. Misfolding can occur due to folding, they can stem from folding intermediates. They can also come from cases where your native protein has undergone some sort of stress that led to transient unfolding, and they misfolded in an attempt to get back to the native state. In these cases, chaperones can come in and try to remodel the misfolded protein such that hopefully you hit that native protein state again. Or via degradation systems, you can take the misfolded protein and just degrade it to peptide fragments. The peptide fragment route is really the last case scenario. It's a last resort because you put a lot of energy into synthesizing a protein. Instead of trying to just basically scratch this thing and start over, it is energetically more efficient to try to refold it first before you degrade. So when we talk about the potential fates here, if we have a misfolded protein, we're more than likely first going to try to take it back to the folding intermediate, which then goes to the native state, before we decide to degrade to peptide fragments. It's just a matter of being efficient with regards to your energy. It takes a lot of ATP, a lot of energy to synthesize a protein in the first place. So instead of just scratching it all and starting over, it makes more sense. It's more economic to try to refold that protein instead of just making it again. Okay, so protein folding there's a lot going on here. And so what I really just want to go back and say again is this top branch, this is ideal. This is the situation where everything goes right. But there's a lot of possibility for things to go wrong. So just try to keep that in mind. Now let's talk denaturation. So I've given you this before, but I'm going to redefine this term for you. And I'm not really redefining it, but rather just reminding you of what I said in previous lectures. So first, what is protein denaturation? It is simply a loss of three-dimensional structure sufficient to cause loss of function for a protein or enzyme. Okay, so that doesn't mean complete loss of structure. That just means loss of structure sufficient to disrupt function. Now, when we look at this figure here, you'll see we go from native state to partially misfolded or unfolded to what is labeled as denatured. In this particular case, complete loss of structure is the denatured state. But imagine a situation where you have an enzyme that loses function once you go to that partially unfolded state. So in those cases, I would classify this as the denatured state. So just remember, at the end of the day, to denature something, all that means is that you have disrupted the three-dimensional structure enough to cause loss of function. That doesn't necessarily mean that you have disrupted all secondary structure and gone to the noodle state that is shown here. That is not what that means. Just to denature is to lose structure to the point that you also lose function. In general, there are two primary ways that denaturation can occur: heat or chemical denaturation. So before I get there, let me lay out a hypothetical scenario for you. Imagine that you are a protein that all you want to do is find your lowest energy state. That's the state that's going to keep you happy. So here, what we are really considering is the unfolded conformation where everything is flexible, your side chains are entropically happy, they're hydrated with water. Cool, everyone is happy. We are comparing that unfolded state to the native state where you do have structure and are marginally more stable than the completely unfolded state. So as I go into this discussion of denaturation, what I want you to try to remember is that ultimately what we're doing, whether it be by heat or chemical treatment, we are changing the thermodynamic landscape for that system. In these cases, what we are doing is making, when we are denaturing something, we are making that denatured state more thermodynamically favorable than the folded native state. So we're basically just providing an alternative lowest energy state that happens to be unstructured. So try to remember that as we talk about this. So as we talk about heat denaturation, most proteins can be killed denatured by heat. If you think about the fact that protein structure is stabilized by a network of non-covalent interactions, where each individual interaction, think about the hydrogen bond, for example, the individual hydrogen bond equates to roughly 1 kcal per mole of free energy. So if I raise the temperature of the system, I actually make it it is very easy to disrupt a hydrogen bond, because as soon as the energy of the system exceeds the favorable energy that is brought by that individual hydrogen bond, that hydrogen bond interaction, it's not so likely to occur anymore. So basically what I'm doing is, if you think about this in terms of non-covalent interactions, by increasing the temperature, I can pretty readily disrupt non-covalent interactions that support protein structure and therefore push the system towards forming more unfolded protein. If you want to think about this in different terms, this really is an equilibrium between unfolded and folded states, and heat pushes that equilibrium back towards the unfolded state. Okay, so there's some interesting nuance that we need to talk about here. If you increase the temperature slowly of a solution of protein, you're going to start disrupting those hydrogen bonding interactions. But, the protein's overall conformation generally will remain intact, and this is due to the hydrophobic effect. That is until you see an abrupt loss of structure. Okay, so let me point you over here on the right-hand side, this plot. These are what we call melting curves. So on the y-axis, you have percent protein unfolded as a function of temperature on the x-axis. So you'll see as I increase the temperature, you see a bit of protein unfolding occur, but you'll reach a certain point where suddenly the amount of protein unfolded shoots up. It's via almost a near vertical line until it levels off once everything has unfolded. So this is what I'm talking about here. You see, as you slowly increase the temperature, everything remains intact until you reach a certain point where it just shoots up. So the shape of this curve, this S shape, this is called a sigmoidal curve. So this is a sigmoidal function. Now, this sudden loss of structure suggests that protein unfolding is what we refer to as cooperative. Okay, so cooperative, what does that mean? Well, what that means is if I disrupt structure in one part of my protein, that ultimately destabilizes other parts of my protein. And so this is, in essence, an all or none event. It's like a domino effect. If I disrupt structure at one part of my protein, that is, a domino effect that destabilizes the rest of my protein, and so it just all sort of falls apart at one point. Now, this is interesting because what we can do is use the sigmoidal shape of this curve to define a midpoint. So if we look at the condition where you have 50% unfolded, so 50% walkover here, and then extrapolate to the x-axis, the corresponding temperature is what we refer to as the melting temperature, or Tm. So the Tm of a protein describes the temperature at which you have 50% folded protein and 50% unfolded protein. Now, the Tm for proteins are going to be case specific. So you'll see here two examples, ribonuclease A and apomyoglobin. They have two different Tms. The Tm for apomyoglobin is higher than for ribonuclease A. So from that, I can infer if my Tm is higher, my protein structure is actually more stable. It takes more temperature, more heat to disrupt my structure. So I can say, based on TMs, that apomyoglobin is more stable than ribonuclease A. The typical explanation for this is that protein stability is directly linked to your non-covalent interactions. So you're probably going to see that apomyoglobin has more, say, hydrogen bonding interactions or disulfide bonds that stabilize it relative to ribonuclease A. To give you an example, many thermophilic enzymes have a lot more hydrogen bonding interactions relative to your mesophilic enzymes that exist at just ambient temperatures. So those thermophilic enzymes tend to be much more stable at higher temperatures relative to your mesophilic enzymes because of additional non-covalent interactions that stabilize the structure of the enzyme or protein. An alternative to heat denaturation is chemical denaturation. Proteins can be readily denatured by exposure to pH extremes, organic solvents, chaotropic agents, as well as detergents. So most of these function by disrupting hydrophobic interactions that make up your nonpolar protein interior. Now, in particular, I will focus on organic solvents and a class of molecules that are called chaotropic agents. So chaotropes are molecules that in water pretty extensively disrupt the hydrogen bonding network between water molecules and your protein. So this is really important because what that means is basically it disrupts, diminishes that entropic cost of that would be associated with the hydrophobic effect. So the hydrophobic effect, this is what causes your protein to adopt a structure with a nonpolar core, and basically you're bearing away all of your nonpolar functional groups from water. You don't want normally that nonpolar material to interact with water because there is a significant entropic penalty to doing so. Now, chaotropes, what they do is disrupt the hydrogen bonding network, which also makes it so that the entropic penalty that would normally be associated with exposure of nonpolar material with water, that goes away. So in essence, what happens is upon exposure to chaotropes, the thermodynamic landscape changes. So suddenly your hydrophobic effect is not the major driver of protein structure anymore, and the protein can more readily fall apart, more or less. You can move back towards the unfolded state due to that change in the thermodynamics that stems from your chaotrope treatment. So common chaotropes are things like guanidine hydrochloride. This should look familiar to you. The side chain of arginine is a guanidinium group, so that is what you're seeing here. Urea is another example of a chaotrope. The behavior looks a lot like heat denaturation. So here on the right-hand side, you'll see another unfolding curve where you have fraction unfolded on the y-axis, and on the x-axis you have concentration of denaturant. This is guanidinium hydrochloride or guanidine hydrochloride. It could equally be urea concentration, but denaturant concentration. As I increase the denaturant concentration, you'll see yet another cooperative or sigmoidal transition, which is exactly what you saw for heat treatment. So yet again, protein unfolding, it is cooperative in nature. It's an all or none event. Whether we treat with chaotropes or organic solvents, detergents, change the pH, you're going to see something that is in nearly all cases sigmoidal in character. An additional thing to consider here is the effect of denaturation on protein solubility. Remember, when a protein is folded into its globular shape, your nonpolar functional groups are buried away from water. The hydrophobic effect is the reason that you would do such a thing, that you would form a structure with a nonpolar interior and a polar exterior. Now, upon denaturation of proteins, you promote an unfolded state, or at least a state that has more exposed nonpolar surfaces relative to the folded state. And what can happen is those nonpolar surfaces that are newly exposed can interact with neighboring molecules to lead to protein aggregates. In some cases, these aggregates will be soluble, but in many cases, they are not soluble. So you'll see, upon unfolding of a protein, irreversible, insoluble protein aggregation. You'll see precipitation occur. A very common example that I hope you are all familiar with is that of the egg. So the dominant protein in an egg is albumin, or ovalbumin. So that's what you're seeing here. This is the structure of ovalbumin. As you cook an egg, you're going to see that your formerly clear, you crack an egg and it's largely clear, but as you start heating it, you see a change in color. Your non-yolk material turns white. So what you're seeing here is actually thermally induced protein aggregation and subsequent precipitation. This is just precipitated ovalbumin here. So denaturation of a protein structure often decreases solubility for that protein in solution, leading to this effect. Because protein three-dimensional structure is really built from a network of non-covalent interactions, what that means is that the denaturation that we're talking about theoretically should be reversible. If you disrupt non-covalent interactions via the introduction of some stressor, theoretically, once you remove that stressor, you should be able to fold the protein back into its native state. Okay, now that's all theory, but what about reality? How does this actually work? Well, this has been shown in early experiments done by Christian Amundsen. He did work with the enzyme ribonuclease A, which is 124 amino acids long. It also has 4 disulfide bonds. The native state is schematically represented here. You'll see 1, 2, 3, and 4 different disulfide bonds. So in his experiment, what he did was he introduced the chaeotrope urea. He also added in mercaptoethanol. So urea is going to induce unfolding of the protein, and basically you're going to denature your protein via the introduction of urea. Mercaptoethanol is a reducing agent that is going to disrupt your disulfide bonds. So after that, the introduction of those two things, you should have an unfolded ribonuclease A. That's what you have right here. All of your individual disulfide bonds have been converted to the reduced free cysteine side chains. So you're completely unfolded here. Now, what happens next is interesting. So what he did was he removed urea and mercaptoethanol after doing this, and what he found was that this unfolded state returned to its native state, complete with correct disulfide linkages, complete with the enzymatic activity that you would expect for the native state. Now, to put this into perspective, the probability that RNase A would properly reform all 4 disulfide bonds after renaturation is 1 in 105. So the probability is quite low that this would occur. Now, why? How could this be? Well, the lesson that was taken away from this experiment, as well as many others, is that the native structure, the tertiary structure for your protein, it is determined solely by the amino acid sequence for your protein. All of your amino acids that are present in the sequence that they are dictate the final structure for your protein. So theoretically, you should be able to denature a protein, unfold it, remove that stressor, and then have it refold to its native state, because each amino acid that is present in your primary sequence is associated with certain functional groups. Those functional groups ultimately lead, through the interactions that they form, lead to the lowest energy state in a thermodynamic sense, which is really what we associate with the native state. So the amino acid sequence, the primary sequence, dictates the native structure. Structure is dictated by the amino acid sequence. Now, I will say that this is not always the case. There are some instances where, as I pointed out for ovalbumin, you form insoluble precipitates that are due to protein aggregation. Once that happens, that's pretty hard to reverse. So for some proteins, unfolding denaturation is reversible, but this theoretically should be true for all proteins, but in some instances, this is not actually what happens, just due to reality.
Okay, so this marks the end of part 6 of chapter 4. Today, what I have tried to do here is to really introduce you to the non-covalent and covalent interactions that stabilize protein structure and give you some sense of the order of events as well as the thermodynamic reversibility or irreversibility related to protein folding. Next time, we're going to talk about protein folding and unfolding in a little bit more detail and talk about the physiologic nuance of chaperones and how they work. So that'll be the goal of next time.